papers. He's the author of 2 books, A Guide to Research Techniques in Neuroscience and Designing Science Presentations. So he has a high bar to creating this presentation, which is some kind of a visual guide to figures, paper slides, posters, and more. So we'll be very glad to see that.
Better not suck.
And he does say in our correspondence, maybe this is from the back of one of his books, he lives in Williamstown with his wife and 2 children, where he likes to sleep in and take naps whenever possible. And I, as a professor and as a student before that, like to take naps whenever possible, especially during lectures. So if you see me in the lecture, it's on purpose because of the topic.
Great, thank you. Thank you, Jay, for the introduction. I want to thank you and Lois and our Sigma Xi community for asking me to give this talk. Giving the Sigma Xi talk is the closest I'll get to being in a fraternity, so thank you for this honor. I want to thank Alicia Romack, I think she's around here somewhere, for the kick butt advertising she did around campus. And I want to thank all of you guys for being here. As Jay was saying, if you fall asleep, this is the perfect thing. I'll just think you're paying attention if you fall asleep. Giving a talk about sleep 2/3 of the way through a semester on a Thursday is sort of an ambitious thing to do. So hopefully everybody will learn something today about the sleep deprivation you might be feeling at this very moment. And I hope everybody learns something today. So I want to talk on the subject of a good night's sleep. And this doesn't sound like a very scientific term, but it's a term we use pretty often in our lives. It's not uncommon to wish each other a good night's sleep. Usually if somebody's going to sleep, we say sleep well, good night, and we tell somebody if they look stressed out, if they look like they're having a bad day, we say, oh, you know what you need? You need a good night's sleep. Or if somebody's taking a test the next day, if they're taking the MCAT, or they've got the big game coming up against Wesleyan, we say like, oh, you know what you need? You need a good night's sleep. And then in the morning, we sometimes ask for a report about how you sleep. It's like, oh, how did you sleep? Did you sleep well? It's like, tell me about it. Did you get a good night's sleep? And when you think about it, if you ask somebody if they got a good night's sleep, in some ways that's kind of a ridiculous question because sleep is explicitly defined as being unconscious. So, if you say like, oh, how'd you sleep? Really, the best answer you could give is, I don't know. But in some ways, though, it's a perfectly fine question to ask because I think instinctually, when you wake up in the morning, you can kind of tell if you got a good night's sleep or not. Sometimes you wake up and you say, I just got a great night's sleep, even though you might not know why. And the interesting thing about that, too, is it's not necessarily correlated with how long you slept. So sometimes you can sleep for 9 hours, 10 hours, and you wake up and you still feel like you're tired, and you wonder why. It's like, I just got 10 hours of sleep. Why do I feel so tired? So my goal today is to try to scientifically define what a good night's sleep means from a neuroscience point of view. And I'm going to do this in 3 acts, essentially. First, I'm going to talk about what sleep is. And so I like to see my son Liam sleeping a lot of the time. So here he is passed out. So we'll talk about what's going on in Liam's brain and body while he's sleeping and what's going on in our bodies when we sleep at night. And then I want to go and talk a little bit about how sleep is regulated. Sleep turns out to be pretty much a brain phenomenon. The body is very active during sleep as well, and there's a lot of important things that happen in the body, but it's all ultimately controlled by the brain, which is why it's such an important neuroscience topic. So I want to talk about how sleep is regulated, and this is going to give me an opportunity to talk about my research a little bit and what my lab is doing here at Williams College. And then finally, at the end, I don't have very much to say on this topic. Hopefully it's not the main reason everybody came, but how can we sleep better? And I thought that I would highlight this by one of the worst pictures of me ever taken. So, yeah, that's me. Thank you to my wife for snapping that picture while I was asleep. So I'll start out just by defining what is sleep. It's kind of funny that it's actually kind of hard to define exactly what sleep is. And we can look at animals while they're sleeping or people while they're sleeping, and you can say, okay, well, clearly this dog looks like it's asleep, our cats look like they're asleep, and you can tell when your friends are asleep. But how can we actually define exactly if this dog is asleep or not? How do we know that this dog isn't in a coma or something? And so perhaps the dog is dead. So we have to description of exactly what's going on in our our puppy. And so it turns out actually for neuroscientists and biologists, this is actually a hard question, but it's a really important question when you're talking about different kinds of animals. So for example, one of my friends in graduate school, his name's Tohei Yokogawa, he tried to develop zebrafish as a model to study sleep. And so here we see this fish moving around. And in order to characterize the genetics of sleep in zebrafish, first he had to decide if the fish was sleeping. So you can see this movie here of the fish. And just for a brief moment here, the fish goes down to the bottom of the cage. And now we can ask a question. Is this fish asleep? Maybe the fish is just thinking about its weekend. You never know. Maybe it's just pausing to think about life. But the fish doesn't close its eyes, so how do we know if it's sleeping or not? So what Tohei had to do in order to characterize sleep in zebrafish was to use these standard definitions of sleep that neuroscientists use. And there's a few criteria that we use to determine if an animal is indeed sleeping or not. So when we define sleep, one of the big characteristics that I think everybody looks at is whether the animal is immobile. So this is an obvious thing that we look for in each other and we look for in our pets. But during sleep, an animal is typically not moving. And there's a couple exceptions to that. For example, migratory birds, half of their brain actually sleeps while they're migrating and while they're on these long flights. But usually there's this relative immobile period of animals. But what really defines sleep as opposed to a coma or rest or death is some other features. So there's a higher sensory threshold when you're sleeping. Obviously, if somebody taps me on my shoulder, I'll turn to look at them, but if I'm sleeping, it's going to take a much larger tap. But if you reach that threshold, you can still turn and you can awaken if the noise is loud enough or the visual stimulus is loud enough. So here our fish has gotten back to work. There's a stereotyped posture that every animal has, so every species seems to have a posture that we like. So humans, we like to lie down. Giraffes actually don't lie down when they're sleeping for maybe obvious reasons. But every animal has its own posture. Sleep is obviously reversible, which is what separates it from a coma or death. So you can wake an animal up. If you can't wake the animal up, then chances are there's something wrong with it, besides the fact that it's sleeping. And then finally, and this is a big reason that you can tell something's sleeping, is that sleep is homeostatic. And what I mean by that is that the more you don't sleep, the more you want to sleep. And in that sense, it puts it in the same category as thirst or food intake. The longer you go without water, the more you want water. The longer you go without food, the more you want food. So if Tohei went in and basically didn't let this animal have a chance to sleep and kept knocking the fish around every time it tried to fall asleep, the fish would try to make up for it later, and it would be much easier for the fish to fall asleep. And so this is how we know it's something very consistent among species. So this is sort of a behavioral definition of sleep, but neuroscientists in vertebrates and in mammals can actually define this a lot better using a technology called electroencephalography. This is called EEG, and you may have heard about this before, but what EEG does is you put these electrodes on the surface of the scalp. It's not invasive, so anyone can sign up for it and do it. And what these electrodes are measuring is below the scalp, below the skull, there's neural activity of millions of neurons going on, and these electrodes can pick them up and actually can diagnose exactly what's going wrong. So when you were entering, maybe you noticed this little EEG wave I've got playing. This was a recording from a mouse. So here we've got EEG, and then there's electromyography as well. Yeah, that's right, I've got 2 PowerPoint presentations. We've got EEG over here, EMG on the bottom, and you might wonder, why is this useful? This looks like just a bunch of squiggly lines, and it's hard to tell what we're looking at here. And a good metaphor I like to have about EEG is if you imagine a group of people. So for example, Times Square on New Year's Eve. And you imagine, what would it be like if you put a microphone above Times Square on New Year's Eve? So what you would pick up is essentially a million conversations. You'd hear all these individual conversations at once. And that's basically what our EEG is doing. It's picking up the chatter from a million different neurons. But the reason this is useful is because sometimes, for example, on New Year's Eve, the people will synchronize, and their voices will synchronize. So for example, 10 seconds to midnight, people will go 10, 9, 8, and all of a sudden your microphone would pick out that something is happening. Something is different than just this random chatter. And that's exactly what EEG does. EEG is basically able to differentiate between different brain states based on what all the neurons are doing at once. And it turns out that when we're awake, our neurons are relatively desynchronized. We're sort of just like a million conversations at once. So our cortical neurons, they're all just sort of blazing and they're randomly going in terms of their frequency isn't very stereotyped. But when we start to fall asleep, all of a sudden these random conversations among the neurons start fluctuating. and all of a sudden we have this rhythmic wave of activity. And you can pick that up using EEG. So if we look at sort of a typical trace of different EEG patterns from awake, there's 4 stages of what's called non-rapid eye movement sleep, stage 1, 2, 3, and 4. And then you've probably heard of rapid eye movement sleep. And each one of these stages has a different frequency associated with it on the EEG. So the awake state you can kind of think of as sort of a baseline. So what we're picking up here, again, is just random electrical activity. It's just how many of these neurons are firing at once. And you can see that as you go deeper and deeper into sleep, these neurons are starting to synchronize and the amplitude of the waves are starting to fire together. So it's all of a sudden, now the neurons are synchronizing and they're going and they're firing all at once rather than sort of this random chatter when we're awake. And so, in terms of what's actually going on during a night of sleep, basically I just wanted to walk through what might be happening in Liam's brain or your brain as you start to go to sleep. And so we can graph this as a hypnogram and look at the 7 hours of a typical night of sleep, starting with the awake state, and just basically ask what's happening in our brains during these events. So when we start out awake, we do different things before we go to bed. Some of you might fall asleep on the couch or on a chair in the library. Some of you might have a more elaborate nighttime routine. But I think it's kind of interesting that no matter what our routine is before we go to bed, it's really impossible to say when we fell asleep. There's sort of this gray, unmarked border in terms of wakefulness and sleep. But typically what happens as we start to go to sleep is we start to assume this posture, as you guys all know, and we start to slump a little bit. Our breathing slows down, our heart rate slows down, our thoughts scatter. So if we had a singular train of thought and we were focused on something, all of a sudden now different ideas start to blend together and it's harder to focus on something. And we generally just sort of drift into an unconscious state. And it's very hard to say where that unconsciousness started, and it's even hard to see it on the EEG. It just sort of kind of gently goes into that. But eventually what we do, is drift from an awake state into what's known as stage 1 of sleep. And stage 1 of sleep is just the lightest form of sleep that you can have. And in fact, if somebody wakes you up during stage 1 sleep, you might not even know you were sleeping. It's one of these states where maybe if you did fall asleep on the couch, maybe you'd open your eyes and you wouldn't even know if you had fallen asleep or not. You might think you did, but you wouldn't know for sure. This is also the stage that people find themselves in when they're at a talk like this. And everybody probably has this, what's going on. It's like everybody does this thing where your eyes start to, the eyelids droop a little bit. Like this has never happened to anybody here, I'm sure. But actually what's happening when you go from the awake state to stage 1 is this neurotransmitter, acetylcholine. The nervous system communicates with the muscles with this transmitter, acetylcholine, and the nervous system actually pumps less acetylcholine to your eyelids, which is why your eyelids feel heavy, is because literally there's less signal trying to keep them open. So your eyelids are drooling, and you play this game with yourself where you're like, okay, I'll just close them for 3 seconds and I'll open them again. Everybody plays this game. But eventually, maybe you close your eyes, and if you're in a situation like this, you do that head bob thing where you're just like, someone saw you, and it's really embarrassing. But you were actually asleep. You were actually entering stage 1 sleep. But the brain sort of figured out that this was not an appropriate venue for you to fall asleep. So you wake up from stage 1 sleep. But generally, once you go into stage 1 sleep, you'd stay there for about 5 minutes. And stage 1 sleep is really just the body's way of preparing you for the rest of the sleep that you'll get later that night. So if you stay in stage 1 sleep for about 5 minutes, then gradually you'll enter stage 2 sleep. And stage 2 sleep is also considered light sleep. Maybe if somebody woke you up, you would actually be able to say that you were asleep, and you would remember that you had fallen asleep in some kind of vague way. But you'd know that you were asleep. And during this stage, again, your breathing slows down, your heart rate slows down, your levels of cortisol drop, your stress hormone drops in the brain, and you assume this posture. If you weren't in a posture like this before, if you were sitting on the couch, this is generally the time when you just sort of slump over and you crash. And so stage 1 and stage 2 are the light forms of sleep, and this is the time when your body is just really preparing itself for what's known as deep sleep. And when people say deep sleep, in a scientific term, they're really talking about stages 3 and 4 of sleep. So stages 3 and 4 of sleep is slow wave sleep or non-REM sleep is really where these brain waves are synchronizing up. they're really just now starting to fire in these deep waves. And you're totally unconscious. If somebody wakes you up during the stage, this is where you're really groggy. This is where, if somebody wakes you up, it might be really hard for them to do so. And you're going to be sort of nonsensical and not even know what's going on when you wake up. So the big question is, what's happening in stage 3 and 4 of sleep? And the answer seems to be all the benefits of getting a great night's sleep for the body typically happen in stage 3 and 4 of sleep. And there's so many of them. And there's just like way too many to list, but I'm just going to list a few of them. So when you're in stage 3 and 4 of sleep, probably one of the big things that people want to happen is the release of growth hormone from the pituitary gland. So you're sleeping and the brain is pumping out growth hormone. This is especially important in kids, and young kids who aren't getting enough sleep. It's really bad because they might be permanently shorter later in life if they're chronically not getting sleep because they don't get enough growth hormone. The growth hormone helps the muscles grow, the bones grow, and it goes to the liver where it helps release insulin like growth factor 1, and this helps the bones grow as well. And this is also a stage where fat is metabolized a lot more too. So for any athletes, athletes who work out during the day or who run during the day, all the benefits of working out, they actually don't happen while you're working out. If somebody's lifting a barbell or something, sometimes people like to look at themselves in the mirror, they lift something and they check themselves out. So what they're checking out is the increase in blood flow to the muscle. But in terms of what's actually growing and when the growth actually happens, it all happens during stage 3 and stage 4 of sleep. So this is why in professional sports teams, they tell the athletes to get as much sleep as possible, because this is when all the benefit is going to come from working out. So the immune system is also very active during stage 3 and stage 4 of sleep. And this is a complicated slide, but it's basically just showing you that the immune system basically functions by releasing these T cells, and the T cells go and it leads to the activation of other immune processes, and these cells go out and they hunt for pathogens. And people who learn about the immune system will know this in much greater detail than I do. But my point is that when you learn about these processes, most of these processes happen much more in stages 3 and 4 of sleep than they do during the day or during other sleep states. And so this is why when people don't typically get a good night's sleep, they might get sick afterwards. And the reason is you get sick is because the immune system is mostly active during stage 3 and 4 of sleep. So it's almost like you're not letting your immune system do its job if you don't go into stage 3 and 4 of sleep. So another big component of sleep, which is near and dear to my heart, is that the things you learn during the day are consolidated while you sleep. And so memories are really strengthened a lot during sleep. So here's just a picture of some neurons. Some are stained green, some are stained red. And some of you may know that memories are usually based on the fact that neurons, their synaptic strengths change during the day. And so if you learn a new fact, then there might be some neurons that recognize that fact. And the association between the synapses of those two neurons will be strengthened. And that mostly happens during stage 3, stage 4, and REM sleep as well. So a lot of this memory consolidation happens while you're sleeping. And importantly, you forget a lot while you sleep. Sometimes people forget that side of the coin, is that you want to remember things that are useful to you, but you want to forget things that are not useful to you. So if I say, today's Thursday, if I ask you, what were you wearing last Thursday? You don't need to know that. I'm guessing you probably forget what you were wearing last Thursday, and that's a good thing. You don't want to remember all these things because you want to use your brain space in better ways. So you do a lot of forgetting during sleep, and you do a lot of remembering and strengthening of synapses while you're sleeping. And this is a very active process in the brain, and it basically goes through a lot of metabolites. There's a lot of biochemistry that's going on, and there's a lot of waste products that build up. And it builds up during the day, but it especially builds up during this consolidation process. And so the brain has a mechanism for cleaning itself. There's a way for the brain to give itself a bath. And there's, as some of you may know, there's a ventricular system in the brain where essentially there's this liquid called cerebrospinal fluid is produced. And the cerebrospinal fluid is made in the middle of the brain, and it continuously pumps out this fluid all throughout the extracellular space between the neurons. And so it's really washing away the metabolites. And a paper last year, for the very first time in science, basically tried to visualize the washing of the brain. And they put in this fluorescent dye into the ventricular system, and they said, they looked at when is this washing most active. And it turns out that the washing is mostly active when the brain is asleep. So here's this red dye that they put in the asleep brain and a green dye in the awake brain. And what you can see is essentially the asleep brain, this is much more active. Let me just turn down the lights just a little bit more. So you see more red in the asleep brain than you do green in the awake brain. And this is representative of the fact that the brain is literally clearing the mind. So when you wake up and you're like, oh, I got a good night's sleep, my mind just feels clear. Really, that's literally what happened, is that you're washing away all the metabolites and waste products during stages 3 and 4 of sleep that would build up otherwise. And so I like to think of this in some ways as sort of like the brain doing its dirty dishes. In a way, it's, you know, if you think of, you know, if you didn't do your dishes one day of the week on a Monday, that would maybe be okay, maybe you get away with that, but by Friday, a week's worth of dishes would really mean that you just couldn't function in your kitchen anymore. And I think of stages 3 and 4 of sleep and REM sleep as really the brain processing, getting rid of old memories, and washing itself clean. And when you don't get enough sleep, basically this washing process doesn't happen, and you just, that's why the next day it's really harder to just think about things and process things, as the brain's literally not as clean as it should be. It's pretty interesting. So then something totally fascinating happens after stages 3 and 4 of sleep. You leave stage 3 and 4 of sleep, and the EEG takes on a really new state, which people, before they called it rapid eye movement sleep, they actually called it paradoxical sleep. And they called it paradoxical sleep because the EEG looks more similar to wakefulness than it does to any other sleep stage. And so people thought, well, that's interesting, the EEG looks like the person's awake even though they're clearly asleep. And then people noticed that the eyes are moving. So the body is totally paralyzed during this state, but if you look at somebody's eyes, their eyes are moving back and forth very rapidly. And you can see this in your pets too. Like some of you probably see your dog sleeping, and every now and then when you see your dog sleeping, you can see their eyes moving around. So that's your dog dreaming. And the interesting thing is the eye is the only muscles that move around. And during REM sleep, there's a lot of replay of the day's events. And it's kind of a mystery what dreams are and exactly what the brain is doing during REM sleep. It's definitely consolidating a lot of memories. But if people record from mice or birds, and they record the kinds of things they're doing during the day. For example, if you put a rat in a maze and you teach the rat how to go through the maze, and you realize that there are certain neurons that fire in a really specific pattern during the day. So basically these neurons are indicating its place in the maze and how to get through the maze. If you let the animal go to sleep at night and then it enters REM sleep, you see that exact same pattern of neuron firing again and again. So it's almost like it's replaying all the memories that happened during the day. And you can actually see that based on the electrophysiology you're looking at. So the first time you go into REM sleep, it doesn't last very long, maybe 10 minutes. But this is really just one complete cycle of sleep. And so the first cycle of sleep maybe lasts an hour and a half, 90 minutes. And then you go and you start this again, and you go into a different cycle, and you go through these cycles maybe 3 or 4 times a night until eventually you get longer and longer periods of REM sleep. You don't get as much stage 3 and 4 of sleep. You go back and forth between REM sleep and maybe stage 2 of sleep, this light sleep. If somebody wakes you up during REM sleep, you're likely to remember your dream. If somebody wakes you up during stage 2 of sleep, you're likely to, it's a little easier to wake you up. And so some people have made these fancy alarm clocks where you strap something to your wrist and it tries to predict whether or not you're in stage 2 of sleep or REM sleep. And you can actually have a plus or minus window for when you want to wake up. So you can say, I'd like to wake up at 7:30, but if I'm in the middle of REM sleep, wake me up at 7:45. And the idea is that you'll wake up a little bit easier if you do that. So this is just one way to try to harness the EEG power. But obviously people can't afford a $30,000 EEG setup, and these little things you wear around your wrist are sometimes not as accurate, but they're fun. But essentially, you go through these stages, and then if you don't have an alarm clock, or if you do have an alarm clock, or you have a kid who likes to wake you up and play trains, then you wake up in the morning, you turn the lights on. It's just sort of like, okay, it's morning. The light hits your eyes, and you're ready for a brand new day. So this is a full sleep cycle, and the things that happen during sleep. And obviously, with all these benefits of the things that are happening during sleep, if you don't get enough, then obviously you're sleep deprived, and the symptoms of sleep deprivation is essentially not benefiting from all of the things I just told you. And so this is why when somebody's sleep deprived, it's harder to remember things, you get sick easier, your muscle starts to waste away a little bit, it's easier to accumulate fat. All of these physiological processes that happen in the body, if you're missing out on sleep, then obviously when you're sleep deprived, you don't get those benefits. But actually from a scientific point of view, sleep deprivation is actually a term that's used in common culture. Everybody knows what sleep deprivation means. This is actually not the term that a lot of sleep scientists like to use to describe getting a lack of sleep. The term that people describe sleep deprivation with in the scientific literature for sleep is actually sleep debt. And it's a slightly different term, but I think it's better. And it's more descriptive because sleep deprivation implies maybe an acute lack of sleep. So maybe on a Monday, if you normally get 7 hours of sleep, and that's good for you, if you got 5 hours of sleep, you can say, okay, I was a little sleep deprived that night. But sleep debt refers to the continuous accumulation of not getting enough sleep. If you've not gotten enough sleep, you know, if you only get 3 hours of sleep or 4 hours of sleep continuously over a week, that's not really just sleep deprivation anymore. That's sleep debt. That's like, you know, not having enough money to pay for lunch one day versus having massive credit card debt. It's essentially the buildup of everything that happens. And the amazing thing about sleep debt, and I really can't believe this is true, this is something that I still think is amazing just to this day, is that the body remembers how much sleep you need. So that if you maybe are 3 hours short of sleep on a Monday, but you get enough sleep on a Tuesday, Wednesday, and Thursday, on Friday you'll still be feeling the effects of the 3 hours of sleep you lost on the Monday. And that's amazing. And a lot of people, they say, oh, I don't know what's wrong. I've been getting plenty of sleep the last few days. But then you remind them, yes, but you just went through the fall semester. So maybe you actually should spend your winter time sleeping in a little bit to pay off your sleep debt. And it's a lot like credit card debt. You can pay it off in little amounts. And just like credit card debt, you can't pay it forward. So you can't say, oh, next week's going to be really hard, so I'm going to get 10 hours of sleep every day. It doesn't work forward. But it definitely works in reverse. And so the effects of not getting enough sleep and having too much sleep debt, they're kind of obvious. It's basically you've got these psychological deficits, you've got poor immune system function, high blood pressure, risk of heart disease, the muscles don't grow enough, and there's so many papers that have described this. There's lots of different studies, you know, and by lots I mean thousands of studies. I've never seen a single paper that shows that sleep deprivation is good for anything, or sleep debt is good for anything. But there's thousands of studies that show that the problem. So, if, you know, obviously here's the link between sleep quantity and academic performance for the college student. I'll spoil the ending. It hurts academic performance. Here's a neat paper that was from Nature. Sleep inspires insight. So they gave people these tasks to do that required a creative solution. And people who were even lightly sleep deprived had a harder time figuring out the solution than people who had gotten a normal night's sleep. Here's one from Neuron. That's a good neuroscience journal. Practice with sleep makes perfect sleep-dependent motor skill learning. So when people think about learning, usually you think of declarative memory, but actually the body has a lot of learning too. If you learn to play a sport or you're practicing a sport, that's really learning as well. And this learning also is consolidated during sleep, and so this is just going to show you that sleep enhances that process. This is a neat study I like. I used to do these outreach talks in high school when I lived in Seattle, and I'd go to schools sometimes, or I'd talk to teachers, and sometimes middle school students and high school students, they'd be like, yeah, I know we need a lot of sleep. But then I'd show them this, and they'd be like, oh, they'd start to pay attention. They're like, oh, attractiveness is correlated with sleep. What this study did was they showed two pictures of the same person, and in one of the pictures was the person's very sleep deprived, and the other one was relatively not sleep deprived. And the photographer had no idea how much sleep the person had. So they tried to control for, you know, the fact that maybe somebody walked in and, you know, was like looking very tired. But the photographer was basically told, just take a picture of their face, and then the people judge these pictures. They say, okay, which version of this guy do you think is the most attractive? And the answer actually, it correlates very well. So people think that the less tired the person is, the more attractive they are. And they also rate them as looking healthier. They say, oh, that person just looks healthy. And they were able to discriminate between the two of them. And so this is just to show you, not only will you be healthier, but you'll look healthier, and you'll get better replies on yip yap. So, that was controversial. All right, so local sleep in a weight crash. This is one of my favorite papers ever. This paper came out just a few years ago, and I'm surprised nobody had done what this paper had done before. What this paper did was essentially take a rat and sleep deprive it, and instead of using EEG, they put in these tiny little electrodes in different spots of its brain. And this is actually not incredibly hard to do, and I was really surprised nobody tried recording from single brain regions at once rather than using EEG. But the amazing thing about this paper, what they found was that when a rat is sleep deprived, even though it's awake and it's moving around and the EEG looks like wakefulness as it does over here, the amazing thing is that parts of its brain were falling asleep. And parts of its brain, if you looked at a tiny region of the brain, would look like non-REM sleep. And this would happen for a couple seconds each. It's basically different parts of the cortex took turns going offline. And what's amazing about this is just the fact that, I'm looking at all of you and you all look awake. What's amazing, maybe for those of you who are very sleep deprived, there are parts of your brain that are actually taking turns falling asleep for you. And you can just imagine, what the deficits that might be causing you, that part of your brain is not fully conscious. It's a neat thing to think you're awake, but maybe you're not 100% conscious. Maybe you're 95% conscious, and it's nothing you could ever measure. It's just it's a really neat concept. So, if you're not asleep, obviously, or if you're not awake and fully paying attention to what's around you, this is obviously very bad, and you can, there's a lot of accidents that can happen, and here's just a fun reporting of some nice statistics, is that sleep deprivation is thought to cause about 1,500 deaths per year. And this is a very hard thing to measure because obviously you can't ask the person who is deceased now, like why they were involved in the accident, but typically this happens with interviews with the people who last saw them, and they say, oh, this person was exhausted and we told them not to drive, and they got in the car and drive anyway. So 1,500 deaths per year is something the media will never report because, saying, oh, we should really do something about people falling asleep, it just doesn't make for a better news story. But you can compare this to a lot of diseases you've heard of, like Ebola, and you can ask, okay, how many people die each year because they're just not getting enough sleep? It's something that really doesn't get talked about. But even in a less severe sense, there's 100,000 car crashes that happen a year that are estimated to be caused because of sleep deprivation. And a lot of disasters that have happened in recent events are thought to have been caused by sleep deprivation, by people not paying attention. So for example, the Challenger disaster, anybody who was alive during the time of the Challenger disaster probably knows that the ultimate cause was determined to be something called O-rings. There were these little O-rings that failed. Something that doesn't get talked about a lot is the people whose job it was to inspect the O-rings had been working a 30-hour shift when it was their time to figure out what was going on. And so perhaps if those people had been sleeping a little bit better, they would have realized that there was something wrong with the O-rings and they could have planned for this. So the Exxon Valdez oil spill, this was caused because the captain had been awake for over 24 hours and he wasn't really paying attention. And then speaking of disasters, even people who support Rick Perry would consider that his debate performances weren't all that good. He famously said, there are 3 federal agencies he would shut down. He just couldn't remember what they were. So in subsequent months, when they did a postmortem on his campaign, he admitted that the night before that debate, he hadn't gotten any sleep. And so maybe if he'd gotten sleep that night, he would have remembered the federal agencies that he wanted to shut down. So it just goes to show you a good night's sleep is definitely worth it. And it's estimated, again, this is hard to estimate, but this one paper in Sleep from a couple years ago tried to estimate in the United States how much money is lost with productivity, and they estimated $63.2 billion, although that's obviously a hard thing to estimate as well. But one point I really want to make is that obviously losing out on sleep is tough, and if you don't get the full quantity of sleep that you need, that obviously will lead to deficits. But something that doesn't get reported as much is that sleep quality also matters just as much. And this is why sometimes you can get a full 8 hours of sleep or 10 hours of sleep and you could wake up and you might not feel refreshed. Because it's more than just the time you're asleep, but it's also what your brain was doing while you were asleep. And what I mean by that is, this is the hypnogram I showed you before. So in general, you go and you go through stages 1 through 4 of sleep, you get a little REM sleep, and this cycles throughout the night, and then you wake up. It turns out this is actually a textbook hypnogram. This is something that I can tell you in a sleep talk because it's easy to talk about and it's easy to show. But what a real hypnogram might actually look like in somebody like you and me might be something more like this. Essentially, the body is still going through these sleep stages, but you can see there's sometimes these little awakenings that happen. And the sleep is actually fragmented a little bit, and there's all these inappropriate transitions between the stages of sleep and REM sleep. And this turns out to be just as deleterious as sleep deprivation itself. So you could get a full night of sleep, you can get just the same amount of hours of non-REM sleep and REM sleep, But if your sleep is greatly fragmented like this, it'll really mess up the process of memory consolidation, growth hormone release, and all the things that you benefit from. So the question is, why do you sometimes have this sleep fragmentation, and what causes it, and how can we learn more about it? And this will just bring me to the second part of my talk, is how sleep is regulated, because it's really all about how you break down these sleep states and what is causing the brain to go from one state to another in the first place. And when you know that, then basically you can look and you can ask, okay, well, what's being messed up when we go through these different states? So I want to talk for a little bit about how sleep is regulated. And like I said before, this is a phenomenon of the brain. So there's two systems in the brain that control sleep. And one of them is circadian, so this is the night-dark cycle. This is why we naturally want to feel awake during the day, but feel tired at night. And then there's a homeostatic system as well, where generally by homeostatic again, I mean the less sleep you get, the more you want to sleep. And it's sort of this increased drive to sleep. And sometimes these systems line up. If we're doing a good job with our sleep hygiene, then everything is lining up perfectly. We're awake during the day, and now we want to go to sleep at night. And because we've had a whole day of being awake, the less sleep we're getting, and the more we want to sleep at night. So usually these systems want to be aligned together, but sometimes they're not. Like when you pull an all-nighter, obviously your homeostatic system is telling you to go to bed, but your circadian system is telling you to wake up, and they're in direct conflict with each other. And this is why you can simultaneously sort of get a second wind if somebody pulls an all-nighter. You see the light outside, and you're like, oh, I made it till morning, and you certainly feel a lot more awake than you did before, and that's because your circadian system is coming online. But your homeostatic system is saying, wait a minute, it's time to go to bed, and there's a conflict that's going on in the brain as to whether or not you should go to sleep or not. So just in brief, in how these systems work, the circadian system is regulated by light. So here's an animation from MIT where basically it's showing you that this person is looking at some light, and most of the light we see goes to our visual cortex, and we consciously perceive this as vision. But there's actually some light that never makes it to our conscious visual cortex. There's some signals from the eye that instead of going to visual cortex goes to the hypothalamus. And there's a tiny little group of cells in the hypothalamus here called the suprachiasmatic nucleus. And this is our brain's clock. And this clock is entrained by the light-dark cycle. So the more that you're seeing light, the more the suprachiasmatic nucleus is actually inhibited. And what the suprachiasmatic nucleus does is indirectly projects to another part of the brain called the pineal gland. And the pineal gland releases this hormone called melatonin, which maybe you've heard about. And melatonin helps you to fall asleep a little bit better. It sort of is like a nurturing you to go to sleep. And it stimulates the inhibitory cells of the brain, and it inhibits the excitatory neurons in your brain. So melatonin generally tries to put you to sleep, and the more you see bright light, the more this structure is inhibited, and this is why the day-night cycle is regulated the way it is, and how that works. So it's interesting that the circadian cycle is really just regulated by this one structure here. The homeostatic regulation of sleep is a lot more complicated, and here's a brain, and if I just sort of cut halfway through a brain, this is what it would look like. And just to show you the different parts of the brain that are involved with this, I'm just going to make this into a cartoon. So here's our fake brain. And there's a lot of different cell types that regulate sleep from a homeostatic point of view. So there's a few nuclei, the tubulomammillary nucleus, dorsal raphe nucleus, and the locus coeruleus. And these groups of neurons, they widely project throughout the brain. And they have these unique neurotransmitters. So the tubulomammillary nucleus releases histamine, the dorsal raphe nucleus releases serotonin, and the locus coeruleus releases norepinephrine. And what these neurotransmitters do is they get pumped throughout the entire cortex, and they basically stimulate the cortex and try to drive it into a non-synchronized state. So when these neurons are sort of firing randomly, that's because they're continuously pumped by the three neurotransmitters that are pumped out by these brainstem nuclei down sort of deep in the brain. And then there's a fourth group. There's a group in the hypothalamus that people call hypocretin neurons. And they're called hypocretin neurons because they make this little neuropeptide called hypocretin. And what these neurons do is they also project through the cortex. But what they really do is they project to these three different nuclei here, and they really try to synchronize the activation of all three of these nuclei. So when hypocretin neurons are active, that really tries to drive this wakefulness state. And the sleep disorder narcolepsy, you probably all heard of narcolepsy. People who have narcolepsy have a select degradation of their hypocretin neurons. And so they are basically, it's harder for them to maintain this state of wakefulness. So that's what causes narcolepsy. So on the other side of the coin, you've got these wakefulness nuclei, and then you also have some nuclei that promote sleep. And there's a couple that are well studied, one called melanin concentrating hormone neurons, and another region called the ventrolateral preoptic nucleus. These are inhibitory neurons, and they project to the wake active neurons, and they actively inhibit them. So it's kind of interesting. It's almost like you've got this seesaw in the brain. It's like you've got some neurons that want to keep you awake, you've got some neurons that want to put you to sleep, and they're actively inhibiting each other. And so it's almost like a switch. It's kind of as you go on throughout the day, there's like a seesaw pattern that sort of is driving you to go to sleep. And when you get more sleep at night, the wake active nuclei are sort of like, hey, come on, and then they flip it the other way. And there's sort of this battle going on throughout the 24-hour cycle. So the cool thing about this is that this is the case for human beings, but it's also the case for mice and rats. So we all share this as mammals. We have all these same nuclei, which means we can study this in the lab, and we can actually say which one of these neurons is mostly beneficial for sleep states. Is there something here that plays a greater role for controlling sleep and wakefulness in the brain? And all of the projections are also the same. So you can actually see their anatomical projections and find out that they're also very consistent from one state to another. So one big question in sleep research that's occupied people for the past 5 or 10 years is how do individual populations of neurons influence these sleep-wake states? And how can their inappropriate activity affect sleep fragmentation? So the way this has typically been studied in the past is usually people would try to go down and activate one of these neurons electrically. So you could put a little electrode into the brain and stimulate it and zap it and try to see the effects of stimulating these nuclei in the brain. Or you could go down and try to make a little lesion and look at the effects that way. But it becomes very hard when you have a situation like this, where you have hypercretin neurons right next to melanin concentrating hormone neurons. How would you go down and selectively stimulate just one of those? And to make that point even more, here's a couple histological images. So here are two images where hypercretin is stained for in green, melanin concentrating hormone is stained for in red. And you can see these neurons are right next to each other. They form the same population. So experimentally, for many, many years, it was really hard to say, okay, let's just try to stimulate hypercretin neurons and see if that really does cause animals to wake up more and generate this sleep fragmentation. So this technique that's come around, which some of you know about and have heard from my other talks that I've used in recent years, is something called optogenetics. And this has really revolutionized sleep research. This is the ability to control a very specific population of neurons with light. And the ability to use light means that it's a very sophisticated temporal control that my lab and my lab in graduate school as a postdoc has used now to understand sleep circuits in the brain and the effect of sleep fragmentation. So to give you just a little brief explanation of what this is all about. So this is called Chlamydomonas reynardi. This is a little green algae. And this algae has evolved a little protein on its outer membrane here called channel rhodopsin 2. And this protein is very interesting because it allows ions to pass through it in response to light. So when blue light hits this channel, it'll let cations through. And this is very useful to neuroscientists because, as you may know, neurons are basically all regulated by their ion channels. And when you have more ions passing through the membrane, that means that a neuron will become depolarized and fire an action potential, or hyperpolarized and not fire an action potential. So what neuroscientists have done for about 10 years is take that channel rhodopsin 2 gene and actually put it into neurons. And if you put it into just the neurons you're interested in, just like hypercretin neurons, then you stimulate those neurons with light, then you can figure out exactly what just those neurons are doing. So for example, just the hypercretin neurons and not the melanin concentrating hormone neurons. And so this has proved to be a very effective technique for a lot of neuroscience studies. So there's two main proteins and genes that people use. One is channel rhodopsin 2 from this green algae, and another one, halorhodopsin, actually inhibits the neuron. So channel rhodopsin 2 will stimulate the neuron with light, so if you shine blue light onto these neurons, then it'll allow these ions to go through and cause the activation of the neuron. Or if you have yellow light, this will stimulate this protein and it'll pump chloride ions through the membrane and it will inhibit the neuron. And so I'm not going to talk about this technique in very much detail, but the key thing is that you can really just stimulate or inhibit whatever neurons you're interested in and look at the effects on behavior. So the way we've done this in the lab is to take an animal, in this case a mouse or a rat, and you go to the brain region that you're interested in, and we perform a stereotaxic surgery. So we'll go into the mouse, we'll put on a little cannula, and during a surgical procedure where the mouse or the rat survives, we'll go into the specific part of the brain that we're interested in. So, for example, if we want to target hypocretin neurons, we can inject a little needle into the mouse's brain, and through this needle we can deliver the DNA for channel rhodopsin 2. And typically we do this using a virus that you can just think of as a little DNA delivery machine. So we'll infect this area with the virus, and then after about a week, the cells that we're interested in, the hypocretin neurons, can fluoresce a different color and express the channel rhodopsin transgene. And then later in a behavioral experiment, we can stimulate these neurons however we want to. So if we want to stimulate them, at a certain frequency, we'll just shine blue light on those neurons, and then we'll be able to see, is that really fragmenting sleep, like we predicted well. So the initial experiments we did in my lab when I was at graduate school, we had this mouse, and we could deliver blue light to the mouse. And this is a little EEG that's recording its brain waves, and here's the real-time recording. So it's in non-REM sleep, or slow-wave sleep. And so we've just stimulated hypocretin neurons with blue light at a specific frequency. And sometimes it happens right away, sometimes it takes a little bit, but the animal will basically have this little brief awakening. And typically it'll go right back to sleep. It's something that's basically duplicating one of these sleep fragmentation effects. And so it can basically stimulate the animal into an awake state, but typically it falls right back asleep, unless we leave the blue light on, in which case it keeps the animal awake for a longer time. So the cool thing about this is this gave us a really cool way to study sleep fragmentation. We could say, okay, does sleep fragmentation really negatively affect learning and memory? And the way we could do this is to stimulate hypocretin neurons for a little while, induce the sleep fragmentation, and then ask, what are the behavioral effects of the mouse after the mouse wakes up? And it turns out that what was predicted actually happened. So if you teach a mouse to do something, like for example, this is something called the novel object test. So you expose the mouse to two objects, and the mouse eventually will get bored of the object that it previously encountered. They don't like to explore objects they've already seen. So if you do this, but you fragment their sleep after they've been exposed to these novel objects, it turns out that if you continuously wake up the mouse every 30 seconds by stimulating their hypocretin neurons and you fragment sleep, the mouse will act as if it forgot about the novel object, and it basically doesn't remember the object that it encountered the previous day. So this was a nice demonstration that fragmenting sleep actually did cause these problems in sleep, just like we thought they did. So another thing we looked at were the important downstream targets of hypocretin neurons. So I mentioned before the hypocretin neurons project to these three different wake-promoting nuclei. So we knew that if you stimulated hypocretin neurons, you could wake the animals up. What we didn't know is which one of these neurons was most important to wake up. So what we tried doing was stimulating hypocretin neurons while we inhibited these downstream neurons selectively to say, is one of these more important than the others to keep the animal awake? And the punchline for this, I'll skip a lot of experiments that didn't work, was we found that if we stimulated hypocretin neurons while we inhibited locus ceruleus neurons, that the animals didn't wake up anymore. And so we knew that this projection to the locus ceruleus was very important. So the way we did this essentially, the first thing we did was demonstrate that if you stimulated the locus ceruleus at all, that the animal would wake up. And so we sort of reproduced this idea. Here's the animal sleeping, here's the EEG and the EMG. We would stimulate with channel rhodopsin in the locus ceruleus, sometimes with a very low frequency, but you can see the animal, unlike hypocretin, would wake up almost immediately. It was a very sensitive area, pumping norepinephrine through the brain. So our big thought was that if hypocretin neurons project to the locus ceruleus, we could ask, is activity in these neurons necessary for hypocretin's ability to wake the animal up? So we did this by stimulating the hypocretin neurons at the exact same time we inhibited the locus ceruleus neurons. And this was probably the most technically challenging thing that we've done. And basically you're delivering light to two areas. But the mice, at first they would wake up with this new implant and they'd get used to it. But after a few days, I think this was in some ways sort of like a kid getting braces. It's like the first few days you feel it, but after a few days the mice just got used to this. And they could tell that they could move freely about their cage. So what we do is basically stimulate the hypercretin neurons and look for sleep fragmentation, but at the same time we stimulated hypercretin neurons, we would inhibit the locus coeruleus. And we asked what happened. And so here's sort of a race. Here's in a few seconds you'll see the blue light going into these two sleeping mice. And in the one on the right, there's going to be simultaneous yellow light inhibiting. So here we've got this light show going on. During a real experiment, we would actually cover the fibers so that the mice wouldn't have any opportunity to see the light. But here we're just making it visible for these movies. So these are representative examples. And when there's no modulation of the LC, the hypercretin neurons essentially look just like they did before. You get this brief wakefulness event, sometimes immediately, sometimes within 10 or 20 seconds. But when the LC was inhibited, this stops the hypercretin neurons from being able to wake up the animal, and it showed you that this pathway was really necessary. And so these are the ways that my lab and other sleep labs are basically trying to dissect the effects of these neurons on sleep fragmentation. And one kind of neat anecdote of this, I couldn't wait, this is like my first big result as a graduate student, and I couldn't wait to present this at the annual Society for Neuroscience meeting that happens every year. And it occurred in Chicago the year that I was done with this. And essentially, I got off the plane, I'd never been to Chicago before, and I saw these signs everywhere. I love LC, I love LC, I love LC. And I was like looking for something on these signs, like what is this? And I thought it was really funny because I was like talking all about the LC. And I had no idea what this was, and for like days I had no idea. And I asked people, like, what is this? And they didn't know either. And it turns out I was a little disappointed. These are ads for lens crafters. I thought this was, but for a few while I just thought they were celebrating what it was I'd done. But essentially, this is sort of the current state of sleep research, is people are going in and they're selectively trying to activate these different neurons and look at the effects of some of these neurons on other neurons. And to this date, a lot of people have stimulated individual populations of these neurons, and every single one of these neurons has now been stimulated with channel rhodopsin. And these hypotheses of what they're doing has proven true, and they promote wakefulness or promote sleep in very different ways. So I'm continuing this research here at Williams College. And one of the first things that I've done is actually start to look at hypercretin neurons and try to dissect them a little bit more anatomically than I had done before. And when I got here last year, one of the first questions that I was interested in is, are these populations of neurons, is it really that every hypercretin neuron projects to every single downstream region? Or are there some hypercretin neurons that only project some places and some that only project other places? And the first person who worked on this was one of my thesis students last year. So here's my two thesis students, Allie Braidner and Monacy Iyer. And Monacy had this as her project. Allie had a cool project looking at thirst, actually. But Monacy was basically interested in, are there two functional subdivisions of populations of hypercretin neurons? And so what she did was she looked at two downstream regions, and what she would do is she would inject a little tracer into one of these regions. So maybe she put a green retrograde tracer in the locus coeruleus and a red retrograde tracer in the tubomammillary nucleus. And what these tracers do is they go backwards to the cells that project to them. So you inject something in the LC, and then the green signal will wind up in the cell bodies of the neurons that project to them. So if she injected something green into the locus coeruleus and something red into the tubomammillary nucleus, and then she stained hypercretin neurons in blue, she could look at the blue neurons and she could say, are there blue neurons that express green and red, or are they only expressing green, or are they only expressing red? And she did this in a lot of different downstream regions and a lot of different combinations. And with some of these, she basically found, like for example, here's one of her studies. So here's this, the blue hypercretin neurons. Sometimes the green tracer and the red tracer showed absolutely no overlap. So basically she can conclude that these neurons were completely different and that they projected to two completely different areas. But sometimes she found that the green and the red neurons completely overlapped in a very statistically significant way. And so what she's basically found is that hypercretin neurons you can essentially divide into two subpopulations. And there are some hypercretin neurons that project to stress areas and arousal areas, and some hypercretin neurons that project to reward areas. And this is sort of the first big thing that we had out of my lab, and we're working on writing this up right now. So the last thing my lab is doing right now to study sleep, essentially we have all these different sleep nuclei, and a lot of different sleep labs are working on these very well characterized sleep nuclei and how they affect the brain. And I thought, because I was here at Williams and I didn't want to really compete with a lot of the labs that were studying these neurons that are already very well known to control sleep and wakefulness. What if we looked at something else and looked at if there's another population of neurons that can also affect sleep fragmentation and sleep states and wake states? And an obvious choice was another homeostatic system. A big question we had was, when you're hungry, maybe that could keep you awake. Or if you wake up in the middle of the night and you're feeling hungry, is that something that could fragment your sleep just as much? And so this is a project that we're beginning right now. And we're asking, how does hunger affect sleep? And the cool thing is that if we use optogenetics to do this, we don't have to food deprive an animal. So if you food deprive an animal, you're also going to reduce the animal's nutrition, you're going to reduce its health because it's not getting the nutrients it needs. But if you just stimulate a hunger area, then that will reproduce the effects of hunger without having to food deprive it. And one brain region, which I studied as a postdoc, are these neurons down here called AGRP neurons, which are the neurons that signal hunger in the brain. So here's a picture of these AGRP neurons down in the very bottom of the hypothalamus. And we actually had some technical issues being able to target these neurons specifically. But over the summer, I worked with a couple really good summer students, Zoe Trupner and Anna Reba, and they basically had this nice poster at the end where they figured out how to target AGRP neurons. And this made it very nice. And this was their beautiful picture at the very end of the summer, these beautiful AGRP neurons that we could now specifically target with the channel rhodopsin protein. So because we're able to target this now, my current thesis students, Kelsey Loy, Nitan Goldstein, and Allison Smith, are now able to target AGRP neurons for their different projects. And doing these surgeries are a little complicated, so first we're starting on oranges, but eventually we're working our way up to mice, and I'm very proud of everybody. We're doing these surgeries very well. And we're asking basically, okay, if you stimulate these AGRP neurons, either while the animal is awake or while the animal is asleep, how is that going to affect their sleep-wake fragmentation when they fall asleep and they go to bed at night? So I'm very curious what the answer to this will be. And it's very exciting to see these sleep circuits actually take shape. Because ultimately, we're not interested in the mice, really. We're really interested in humans. But because, again, all of these nuclei in mice are shared between the mouse brain and the human brain, we really think that we're learning about ourselves when we do this. And we think that this anatomy is really going to help us ultimately learn about the human brain and the way that this is regulated in us. So because we're talking about the human brain, ultimately, I thought I'd just end with one or two slides about how we can sleep better. Now that we know about sleep fragmentation and we know about the kinds of things that control sleep and wakefulness, are there ways that we can use this to get a better night's sleep? And really, the best things that people have offered, I don't have any miracle cures, and I don't want this to be like a self-help talk or anything like that, but people have found that kids sleep a lot better than adults do. And it's really during the teenage years that people start to have bad night's sleep. And the one thing that seems really correlated with a good night's sleep and a bad night's sleep is really developing a nighttime routine and preparing yourself for a good night's sleep. And this is something we stopped doing as teenagers. We usually stop doing it as adults. But it's something that has been shown a lot of times to be really good for states of getting a really good, unfragmented night of sleep. There's a few really specific things you can do to avoid before bed to get a better night's sleep. So one of them is carbohydrates. So if you eat carbohydrates while you're sleeping, that actually affects hypocretin neurons. So hypocretin neurons are stimulated by glucose and by sugars. And so if you stimulate your hypocretin neurons, then obviously this is going to lead to the same sleep fragmentation that we showed in the lab. So alcohol is actually interesting because alcohol is something that sometimes people deliberately take to fall asleep. Sometimes there's people who deliberately have a glass of wine or a nice little nightcap. This is where the term nightcap comes from. And they think, okay, well, I'll just drink something to help calm my worries and I'll go to sleep. And it's true that this actually might help you fall asleep a little faster. The problem is that about an hour or two after you fall asleep, the liver metabolizes alcohol into carbohydrates. And so now you've got more carbohydrates coming. And so even if alcohol helps somebody fall asleep faster, it'll wake people up typically in the middle of the night. And then the last thing I think, and this is a big hot topic these days, I mentioned the circadian system is dependent on light. And the more light you see inhibits your suprachiasmatic nucleus, which inhibits your melatonin system. And yet these days, one of the last things we do before we go to sleep is stare at these bright screens that are just deliberately pumping light into our eyeballs and inhibiting melatonin secretion. So by inhibiting melatonin secretion, it's essentially stopping the melatonin system from keeping us asleep. And so it's another thing about sleep fragmentation. The last big thing to avoid before bed, and this is something that's just easier said than done, and I have no solutions to this, it's been shown time and time again that stress reduces sleep quality. And the more stressed you are, the more difficult it is not just to fall asleep, but you actually wake up a lot more during the night as well. And so the things that people have told me whenever I talk about sleep and things that work for them is sometimes if you are really worried about 5 things in your life, if you just write them down before you go to bed, sometimes people feel that this is a helpful way to sort of get it out of their mind. But this is easier said than done, and I don't promise that this is any sort of cure at all. But if there's one other thing that you can do to help yourself get a better night of sleep, music actually seems to synchronize the EEG waves a little bit. So if you listen to certain kinds of music, this actually helps you enter the slow wave patterns of sleep. So for example, there's like classical music that you can listen to. If you listen to certain sounds, here we go. If you listen to this in sort of a dim room, this will just help you fall asleep a little easier. It's amazing how many people don't listen to music before they fall asleep. Sometimes people have insomnia and they have a harder time falling asleep, but this music can actually kind of soothe you right into sleep. And just the opposite, if you're interested in waking up in the morning, you can, sometimes people have a really hard time waking up, and if you listen to, yeah, exactly like that guy. So, you're proving my point. So essentially, it's amazing. I have a playlist for night, and I have a playlist for day. And sometimes when I'm walking into work, I don't walk in as much as I used to because I drop in my son at daycare, but I used to walk in every day listening to stuff like that, just because it helps get me going just a little bit more. And then finally, people have shown that if you exercise during the day, this will also decrease the time you need to fall asleep. It'll increase your deep sleep and REM sleep, and it'll decrease the fragmentation you experience during sleep. So this is another thing you can do that enhances your sleep state. But essentially, these were the components I wanted to talk about. My big overall message is that a good night's sleep is about sleep quality in addition to sleep quantity. It's not just about how much you get, but how well you do and what you can do to prepare yourself to go to bed. There's this attitude that maybe you don't need sleep. I see posters like this all the time, like no one looks back on their life and remembers the nights they had plenty of sleep. There's people like, I don't need sleep. But it's really, to me, this is just completely missing the point. To me, it's not about your good night of sleep, it's about the day. I don't like to look at this picture of myself. I think I look terrible in this picture. I like to look at this picture of myself. I think I'm much more happy in this picture. It's really not about the night of sleep, it's about awake. It's like a good night's sleep leads to a better day's wake. And so I like to get sleep not for the sake of sleep, but because I like to enjoy myself during the day. So if you want to know any more info about this, there's one book I would recommend. It's called The Promise of Sleep, and it's written by this guy, William Dement. He was the discoverer of REM sleep, and he wrote a really great book about what goes on during your brain during sleep and the reasons to get a good night's sleep. And I would recommend this book above all others for learning about sleep. So I'd just like to thank my students who I've worked with, Allie Greitner, Mossy Iyer, my summer students, Anna Reba, Zoe Trutner for getting the AGRP transduction going, and my current thesis students, Allison Smith, Kelsey Lloyd, Nissan Goldstein. It's one of the best things about being here at Williams to work with people like this, so I'm very appreciative to all of them. I'm appreciative to all of you for being here, and I guess there's a reception, and I'm happy to take any questions. So thank you very much.